Good afternoon, and it's nice to be here. Okay, so the first disclosures, I'm affiliated with a commercial diagnostics company, so be wary of anything I say. All right, so today, in order to understand the scientific basis of this DNA methylation test, we have to understand first what is epigenetics. Epigenetics is not genetics. To understand epigenetics, raise up your right hand right now. Raise up your right hand. Now, interestingly enough, even though we are genetically identical, are different, we're all different genetically from a genotype, our hands are very similar in form and function. Now, but our hands and our brains are genetically identical, but they're very different in form and function. What epigenetics is the software code that comes on top of the DNA code to turn on neurons, turn cells in the brain to be neurons and glia, and those in the hand to form bones and muscle. It's sort of like iPhones. There's all sorts of different chips, all different screens, but iOS makes all iPhones act the same way. Now, at a mechanistic level, it gets deep pretty quick. But for the people in this room, you only have to know one type of epigenetic technology, and that's DNA methylation. And what DNA methylation is very simply talks about the process through which DNA methyltransferases take a methyl group, a CH3 group, from a methyl donor, like folate, stuff you get in greens and things like that, to cytosine residues of DNA. Those which are heavily methylated are turned off. So, and those which are demethylated are turned on. So with the same DNA, epigenase can turn on and turn off the genes for actin and myosin in your hands and GABA decarboxylase in your brain. Now, the fact that epigenetic changes are associated with smoking is old news. In 2008, we published the first gene-specific findings with respect to DNA methylation. And then, after spending a couple $1 million on this experiment, we used a genome-wide approach to show that the most sensitive region of the human epigenome with respect to smoking was in a gene called the aryl hydrocarbon receptor repressor. It's a big word, but it makes perfect sense to everybody in this room, because the most hazardous thing in smoke is, it turns out, smoke, okay? So what we're actually measuring is, this test measures polyaromatic hydrocarbons. Now, polyaromatic hydrocarbons are very toxic, as you all know. And very simply is when you breathe in tobacco smoke, polyaromatic hydrocarbons are absorbed into your lung, cross the alveolar epithelium, into the white blood cells. In the white blood cells, it binds to the aryl hydrocarbon receptor and goes on to turn on cytochromes. And these cytochromes metabolize the polyaromatic hydrocarbons. Very importantly, these cytochromes are absolutely different than the ones that metabolize nicotine. There is no crosstalk between them. So by measuring this, we are specific for smoking. Now, using technology that's been developed since 2012, in 2018, we published this paper that describes the translation of these really hard to do genome-wide tests into a simple test that your standard hospital can use using commercially FDA-approved platforms. This, most recently, we've pushed this technology to the point where we now can definitively show that we can easily classify smokers from non-smokers. And now, like, for instance, as this slide shows, in 400 people that are either females and males that are either current smokers, current smokers, past smokers, or controls. Now, the interesting thing about this, not only does it allow us to classify things extremely reliably, it also allows us to tell how much they're smoking. The more you smoke, the more you demethylate. Light smokers are up around 75%. The more you smoke, the more it goes down. Interesting finding if you're solely interested in detecting smokers. But can this do something more clinically? And the answer is yes, because as smokers change, their risk change. Everybody knows you quit smoking 5 years from now, or today, 5 years from now, your cardiovascular risk is the same as the rest of the population. Now, we can't wait around for 5 years for a lab test to turn positive. But does demethylation occur quick enough that we can use it to guide clinical therapy? And the why is this important? Because smoking is one of the hardest things to do in medicine. And it is one of the most beneficent. It is not only good for the patient, it's good for their families, it's good for their employers, and it's good for their insurance companies. Now, recognizing this, Kevin Volk at the University of Pennsylvania has popularized what's called incentive-based approaches. In other words, as long as everybody benefits, why not pay people to quit? In other words, instead of like, have you quit smoking yet? Hey, here's a positive incentive. Good job. And what he has shown is that using financial incentives to encourage smoking cessation are not only effective, but they're completely combative. Whether you're vaping to quit, you're using the patch, Chantix. Now, the question is could we detect the reversion process quick enough in the smoking cessation process to use it to guide therapy? To determine that, we're in a pilot study with Dr. Eric Hoffman, who's a radiologist in Iowa, and we're taking 80 subjects, and we are paying them to quit. And most importantly is when they quit, they cannot use any nicotine-containing product to quit. So because we're using Nicotine to make sure that they quit smoking. Now, so far, we only have 11 of the 33 first subjects have quit smoking, but that by itself is about 5 to 6 times greater than the usual treatment rates. What do we see with respect to methylation changes? These are the results for light smokers, heavy smokers, and medium smokers. And remember, this is only a few subjects. But what we see is the reversion process is dose dependent. But at the same time, it is fairly rapid. On this axis is the percent change in methylation as a function of time. And on this axis, we see the average methylation changes at 30 days, 60 days, and 90 days after Nicotine verified smoking cessation. And not surprisingly, the more you are down in methylation, the more you bounce back. The light smokers, after 3 months, only bounce back about 3 or 4%. But hey, they were only hanging around 75% anyway. The normal range is 86. But those who are actually at the very lowest, particularly those who are down below 50%, bounce back almost 14% after 3 months. Now, that actually is a very large number because our precision of our test is about 1%. So, in summary today, what we have talked to you a little bit is about cigarette and alcohol consumption, or cigarette consumption, but we also should tell you that we can also measure how much you're drinking as well, using a similar epigenetic technique. These techniques are fast, and we can do them on blood, or we can do them on saliva, equally well. The initial assessments in smoking cessation are very good because they tell you how many cigarettes a person is actually smoking, how heavily they are addicted. Subsequent methylation levels provide an objective measurement of smoking cessation and the quantitative reduction of harm because the amount of reduction is dependent on how much they were smoking. The results, there's no way to fool these results. You know, if we have to, we can verify who gave us the results by genotyping. And for people that are interested in understanding whether people quit smoking with vaping, these are completely compatible with these type of approaches because we're measuring polyaromatic hydrocarbon metabolism, which is not affected by nicotine. And it therefore allows us not only to monitor people during any type of smoking cessation, but to differentiate those who are from chewing tobacco or vaping from those who are actively smoking. And finally, and most importantly as a physician, what this does, it gives us another mechanism through which to empower pro-social behavior, that very pro-social behavior of quitting smoking. Because when a smoker quits, they win, their family wins, the employees win, in fact, everybody wins. So this work, and I just quick word of thanks to my sponsors, the National Institutes of Health and the Foundation for a Smoke-Free World.